Hi, I'm Abraham Mota Gomez, and I'm going to answer my three questions of my argumentative oral exam. Well, in the first question, speaking about any modification that I could make to our proposed process to increase the purity or recovery of the final beta-glucans. Well, our proposed pilot scale process uses only a one-step extraction by alkaline treatment at high pressures after cell rupture by high-pressure homogenization, generating a 78% pure beta-glucan extract. However, no additional purification step is used. So the modification that I could include could be a purification step by precipitation with isopropanol or ethanol to remove, more of the proteins and lipids still present in the final extract, reducing the protein content to 2.8% approximately in the final sample. This type of treatment has already been used at pilot scale for beta-glucan extracted from yeast or also from grains. Additionally, it's essential to comply with the limits required by the legislation, not for the content of beta-glucans that has already reached, but for the content of protein and fat that the final product must have. In the graph, it can be clearly observed how with some purification processes, the protein content of the sample can be reduced considerably, being necessary to be below of the required limit to can be consumed. In the second one, well, the main limitations that exist, I can refer them to four main aspects: the purity generated by the selected methodology, the cost of the whole process, its environmental impact, and from my perspective, being the one with the highest importance, the capacity or possibility of scaling up each operation. All of them, Serotopter, extraction and purification from the laboratory scale to the industrial or pilot scale level. With this in consideration, the improvements to be focused on for a commercial scale process can be reduced in two main considerations. The first one is to select efficiently scalable methodologies in cost-benefit relation, as well as the operation parameters of such methodologies. The second one is these parameters. To scale up an operation to an industrial or commercial level, it's first required to analyze it at laboratory level to determine the best conditions that will generate either the best cell lysis or rupture, recuperation, or purification. These determined variables generally must be maintained constant between the scaling up process. And it's the modification of the operating parameters that will generate that this variable remain constant. A basic example can be the retention time in one spray dryer at lab scale and one spray dryer at industrial scale in the final polish process of the extract of beta-glucans. To be constant, parameters such as airflow rate, chamber volume, or sparging flow needs to be modified to maintain this rotation time constant. And speaking about the last one or the third one, well, beta-glucans are found in the yeast cell wall. So in order to extract them, a unit operation of cell disruption is required. In this case, high pressure homogenization was used because it has been demonstrated that this mechanical operation is feasible to be scaled up to an industrial level. And it's one of the most widely used operation for the disruption of the yeast cell wall. Homogenization is a physical process in which a dispersed system, suspension, or emulsion is forced to flow at a high velocity through a narrow passage, producing a smaller and narrow particle size distribution. In the case of biological samples, such as yeast biomass, the impact of the pressure through the channel generates cell rupture, releasing its intracellular compounds and generating cell wall fragments. Even though high-pressure homogenization is a mechanical method and it's carried out in a non-specific way, it allows maintaining the characteristic of the yeast intracellular components stable. So, for this characteristic, this operation was chosen in our process to generate the lysis of the yeast cell with a pressure of 1,700 bar with 2 passes at the pilot plant scale. I mention this because at laboratory level, we implemented sonication as our cell disruption process. However, our operation par excellence is high-pressure homogenization. These conditions will have a variable named release constant, which needs to be the same in any scalation process that needs to be done to obtain the most similar results from laboratory at the commercial scale process. This cell rupture operation helps to extract the beta-glucans more easily in the next extraction operation, and in the global process is the first unit operation used. So it's really important to have it really well characterized for an efficient extract process of our beta-glucans. And well, that's where my 3 questions, and thanks and goodbye.